Today, we will look at an example from life sciences. The genetic code that we all have in our DNA is a sequence of ATCG letters, which in sets of 3 represent a code to create an amino acid, or represent an amino acid, or translate itself to an amino acid. And these series of amino acids that you can see here, for example, the ASN, GLY, and so forth, these sequence of amino acids would interact with each other like a thread, and they'll basically connect with each other and fold in particular ways that create protein molecules. So here, maybe something like hemoglobin, created out of a series of amino acids represented by what we call as codons, or ATG, or sets of 3 letters in the genetic code. So this is how what you have in your DNA gets converted to something that you can use. For example, collagen is another, powers all much of your structure, like for example hair and stuff, or nails and things like that. So let's try to go more into details of how we can use ZCubes to figure out how can we find out which gene probably it matches to, or which protein it represents, and so forth. So let's do some experiments on that. So here clearly we are saying that genetic code in sets of 3 is representing an amino acid, and this particular mapping we can see in this site here. They have this set of amino acid and codon mapping. So for a set of 3 ATT in your DNA, it represents isoleucine, or GTT represents valine, or ATG represents methionine, and so forth. And there are some special codons, and like TAA, TAG, or TGA, they are called codons. So that means that in the genetic code, whenever this occurs, the transcription will be stopped. And there will also be others called start codon, and so forth. So this set of codons, let's just take it from here and drag and drop it into ZCubes here. So this table is now a part of our space. So if you take your mouse over the table, you can see that it is at the bottom over here in the corner. It'll show that the ID of this table is table 1. So which basically means that if we insert a coding module inside of this, we'll be able to access that table by just simply saying hash table 1. So effectively the table that we have here is being referred to from the coding platform, and you can do this from Calci and so forth, by the way, but essentially you are just accessing the data inside of table 1. Now, given that we have that now, we could say, for example, just get the table and take the columns 3, 1, 0, and 2 and give it to us in that order. So this is the sequence. Basically the table, we are reorienting the columns with the simple tactic. So let's call this as this array that we have as AA codon mapping. So now AA codon mapping will have those values as we are seeing on the screen. Now let's try to somehow make this set of data to another organization where we'll have just one codon and mapping to the rest of the information in this. So what we will do is we will put this code into so that we can take the information from the AO codon mapping table, and for each row in that, we take each row and we split it with commas. So basically we take the first element of each row, that is like ATT, ATC, ATA, and we split it by comma, and then we transpose that array so that we'll have ATT, and then for each of those values, ATT, ATC, ATA, we will extract from that row the values of ATT and then the row 1, row 2, and row 3. So that basically we'll have a table which contains ATT, isoleucine, and I, for example, ILE. Like basically for each of these codon, it'll pick the appropriate amino acid related to that. So, and then we will, here we're just simply printing the A2 codon, but you can see that we are pushing this row of records, row of values that we have created into the A2 codon table. So now A2 codon table has the information from the original table with each codon and its appropriate sets of symbols and amino acid and amino acid code essentially. So now let's do the next step where let's say we have a set of let's simply do a VLOOKUP on the A2 codon table that is above this for the sequence GTA and get us the value from column 4. So this will basically, it's telling us that this is GTA is representing V. So let's see what that means. GTA is representing valine or V, and it has looked up. Now notice that we are using a spreadsheet function here, which is also an inherent part of the ZCubes language. So you can, just like we refer to a table or a calc from the coding aspect of things, we can call any spreadsheet function from the coding language itself, and that's a very, very powerful and differentiating feature of ZCubes. Now that we know that we can look up a codon and find the appropriate amino acid, now means that if we had an arbitrary genetic sequence, so for example, I'll take some genetic sequence from the web, and this is a sample genetic sequence that is generated out of a website called sequence manipulation site, and we have submitted and said, okay, give us random genetic sequences, and it has basically given us this series that we have here. So I'm dragging and dropping it into the ZCubes platform, and this cube, as you can see, has the ID of 6 space, which is shown at the left bottom corner there. So which means that from my coding interface, I could just say Z and 6, which means give me the content of Z6. Now let's get the Z text from that cube 6. However, these have line breaks in them, so we're going to clean it first. So we say, okay, let's just clean this Z text so that all the line breaks disappear. And at this point, we have a random genetic sequence that we would then split with empty string and then split it into chunks of 3. So now, as you can see, we have sets of 3, essentially the codons, from that arbitrary genetic code that we just got from the web. Now we will uppercase that so that it looks like the genetic code. And then for every row that we are seeing here, it's a linear array basically. So we can just say for every row uppercase that, and then for each of those elements that you have there, take each of those cells here and do a VLOOKUP for the value of the cell. So for example, G, look in the table A2 codon and find the 4th column and then give us those values. So it's basically going to give us that these M, S, T, L, and so forth, those are the amino acids that those codons have coded for. So if you join this in one set, we basically have the protein sequence right here. Now imagine that if you have this protein sequence and then you want to find, like you have a protein with a particular, gotten the series of amino acids from the that is derived from this genetic code, we can now take that and for example see if this set of characters have any particular patterns that we can compare with existing proteins. let's assign this to the variable GS and now use another piece of code. If we can find a pattern, so we will do JSDiff on characters on this string that we generated here for a series like this, and where those differences were. And it's telling us that this particular sequence that we were looking for, this might have come from a structure of a protein or so, would have been or could have been created from certain deletions and additions of the original amino acid sequence that we had calculated. So this kind of shows you how we can create a program with information that we are simply dragging and dropping from different websites and whatever, or different data sources. And through simple tactics of managing arrays and computational functions, we are able to do some and solve some serious real-world problems. So that's the session for today, and hope you enjoyed it. Let's know if you have any questions.